grade, I was invited to this biology conference at my school, and I was really excited about attending it because I got to skip all my classes and they gave us free pizza for lunch. Heck yeah! Now, in one of the presentations about the study of diseases, one student from the audience asked, "Why do scientists try to cure rare diseases more than common diseases?" And to that, the lecturer said, "Well, I guess I don't really know. The thing about common diseases is that there are so many factors that influence the final result. Some can include genetics, diet, or maybe something no one knows about yet." And I emphasize yet because that's what science is about, right guys? It's about discovering and testing and innovating, and now, especially during the age of technology, the 21st century, the power of science has been more important than ever. There is a deathly pandemic of SARS-CoV-2 that has taken over our lives and has made life a lot more boring than it usually is. But what if I told you that your life will be able to get back to the normal amount of boring it was? And if your life wasn't that boring during a pandemic, that's up to you. But how? Well, a vaccine is coming, but also a new technology that will be able to detect COVID-19 faster and cheaper. The super cool thing is that this technology that I want to talk to you guys about today is already being retailed across the nation. But let's backtrack and let me introduce you to how it all started with the discovery of CRISPR. Now, many of you might have heard the name CRISPR before. What is it? Not a new Lay's chip flavor, although it definitely should be. CRISPR is short for clustered, regularly interspaced short palindromic repeats. Quite a mouthful, I know, but that's what I'm here for, so let's break it down. According to Quantum magazine, the discovery of CRISPR started in 1987 when a few scientists from Osaka University in Japan found something peculiar. They looked at a specific gene, a single unit of heredity, something we get from both our mom and our dad. And this specific gene, the EAC gene, was from the gut of an E. coli microbe. Microbe being tiny living things that are everywhere. And yes, everywhere, under your bed, in your mouth, and oh, I think you just swallowed one. So when the scientists sequenced the DNA, the genetic material that makes us who we are and its surroundings, they expected to find proteins, molecules that sped up chemical reactions functioning like a light switch. When the light is on, transcription or copying of our body's genetic material into a piece of RNA. A squiggly line, also made out of genetic material that primarily helps make proteins, occurs. But when that light is off, this process stops. However, what the scientists found was that near the EAC gene lay 5 identical segments of DNA, unique spacers, which are 32 random DNA base blocks, so imagine 32 random Lego blocks piling up on each other, and a bunch of genes, which they dubbed cache genes. Now, if anyone likes hamburgers, I know I do. I think I may have an easier way of explaining this concept. Imagine a sandwich filled with 5 patties, which are your short palindromic repeats. And palindromic is like the word race car. Race car is spelled the same forwards and backwards, just like the DNA base blocks, separated with different types of cheese, which is your interspaced part of CRISPR. And let's hope the cheese isn't moldy. And on the top, let's say there's about 4-ish olives, and the more olives, the better. Yeah, unpopular opinion. So after a few years, many more scientists found the same type of genetic sandwiches like the ones in E. coli. So after a bunch of testing and hypothesizing, the function of CRISPR was discovered. Okay, so the $100 question is, what does CRISPR do? What scientists found, according to the Nature journal 33 years ago, was a defense mechanism by microbes. They would capture small pieces of DNA from invading viruses, store that in those interspaced repeats, and if they ever saw that virus again, they'd cut the virus DNA apart, making the bacteria safe. However, in the lab, it's a little different because the purpose is to edit our body's genome, almost like a cut and paste function, but in real life. According to the National Institutes of Health, the 2 main components include Cas9, a DNA cutting protein, and a guide RNA. Together, they locate a specific part of the genome that we're trying to find, unwind that specific target DNA sequence, and from there, they have a few options. They can either cut the nucleotides, the bases of genetic molecules, deactivating the entire gene, block or induce transcription, and transcription, once again, is the copying of our body's genetic material into easier to create proteins molecule. or they can paste or substitute a new nucleotide, which can lead to preventing diseases. According to the Cystic Fibrosis Foundation, one base block error in the CFTR gene can be fixed using CRISPR-Cas9 genome editing. Another disease that will majorly benefit is sickle cell disease, in which once again, only one base is mutated, forming defects in the way blood cells are formed. Alright, let's switch gears and talk about genetic engineering in the context of the problems our world is facing right now. The COVID-19 pandemic has impacted the world on a massive scale and is arguably the most destructive virus of this generation. The cases and deaths have reached new highs, most notably the United States, with a recorded coronavirus death number of around 320,000 people and cases of about 17.7 million people. And these numbers are from the 20th of December, so they have definitely increased. One major problem countries face with a growing infection rate is lack of contact tracers and lack of testing. However, according to Biological Procedures Online, Sherlock, and no, not Sherlock Holmes, although he's pretty cool too, but Sherlock, a genetic engineering solution, will be able to detect COVID-19 using CRISPR-Cas13. Okay, let's recap a little. We already know that CRISPR is the short interspaced repeats in a microorganism, and that Cas9, a DNA cutting protein, is bound to a guide RNA to do so. And once again, that guide helps us find the specific target DNA sequence that we are looking for. Cas13 is a cousin of Cas9 that can be harnessed to actually detect human disease, but probably a really rude cousin because Cas9 is getting all the attention. According to the McGovern Institute, a community of MIT scientists, in microbes and bacteria, Cas13 is also bound to a guide RNA to detect viral RNA. And once that viral RNA is bound, Cas13 starts a process called collateral cleavage, which means it cuts up any RNA segment it detects. Well, doesn't that seem unnecessary? What does cutting up random RNA segments do with anything? Well, surprisingly, this is the meat of Sherlock's capabilities. So, how does Sherlock work? First, researchers take a sample of a patient who possibly has a viral infection. Then they amplify the levels of RNA in that sample and add reporters which are sensitive to Cas13. Next, many engineered Cas13s are added into the sample with a guide RNA to detect that viral RNA. And once that viral RNA is bound, Cas13 starts the process of collateral cleavage. Once again, means it cuts up anything in its path, including the reporters. Well, why the reporters? The reporters being cleaved is what actually detects the virus. Each reporter has a unique label, and when Cas13 cleaves it into two, they create a unique signal, which can help detect whether a person has a virus or not. So imagine a pregnancy test. Well, what does a pregnancy test even do? According to the National Health Service, one early sign of being pregnant is the body's production of the hormone HCG, or human chorionic gonadotropin, in a woman's blood and urine. And when their urine comes in contact with that specially treated strip, the test detects whether or not HCG was detected. Now, this process is very similar to what happens with the cleaved reporters. Once the reporters are sliced, they enter a commercial flow detection system, just like the pregnancy test. And if the sample is negative for that virus, the reporters collect at the first detection line. However, if it's positive, they collect at a different detection line, making a diagnosis very easy to spot, just like the darker red and thinner pink lines on a pregnancy test. Detecting COVID-19 will be significantly easier and will start a path of increased recovery in our pandemic. As I said before, and as I'm saying now, science is amazing. We started with an unknown, unidentifiable gene in a random bacterium and created it into a certified fruit ninja warrior. But instead of cutting fruit, we're cutting genes. Discovery is possible, innovation is always occurring, and solutions will always be created. Okay, so let's answer that student's question. Why do scientists try to cure rare diseases more than common diseases? The lecturer should have said, well, it's because our world is filled with peculiarities that will one day be able to cure the deadliest diseases. But until then, we just gotta wait. So time. Time is the answer to her question, and that will be the answer for our pandemic as well.